This presentation tells the story of three famous scientists who laid the foundation for modern human genetics. This course starts with the assumption that if we want to be able to predict the future, then we need to understand the past. And if we want to understand the origins of the controversies about human genetics, then we need to think about the origins of the subject. And we need to think about the social and cultural assumptions that were held by the scientists and by educated society back then. The story begins in the middle of the 1860s in central London, and also in the small town of Brno, which was then part of the Austro-Hungarian Empire, but which is now part of the Czech Republic. Two worlds which even today are still very different, but back then must have seemed like different universes. In London, the English scientist was Francis Galton, a sophisticated, aristocratic, wealthy English gentleman. Galton was interested in how talent was passed on from parents to children in certain families, and suggested that this transmission could be from both the mother and from the father. This idea he called hereditarianism. Hereditarianism is still a popular worldview today, as we shall discover on this course. Galton first published his views in an important book called Hereditary Genius, written in 1865. In common with other writers of his age, Galton used the term man to refer to humanity or mankind. He argued that in the same way as physical features like height, handspan, weight and face shape can be inherited from parents to children, so can natural abilities such as intelligence and sporting prowess. If this were the case, then it could be relatively easy to improve these characteristics through careful selection of parents, leading to the production of children who were more intelligent and better endowed than their parents. The Victorian era was the age of the successful production of improved farm animals, dogs and horses, through careful selection of parents and breeding. This was called selective breeding. Galton argued that there was little difference between doing this to improve animals and doing it to improve humans. His was a social aim. He wanted to reduce poverty and crime by controlling how poor people could have children. It is important that we realise that from the outset this was an explicit aim of Galton's hereditarianism. The heart of Galton's thinking was also inherently racist. Not all races were considered to be of equal inheritable potential, and not all races were of equal worth. The cultivation and improvement of races through selective breeding was something that Galton termed eugenics. This is the first reference to the term eugenics as a footnote on page 22 of a paper published in 1862. It's important to realise that the methodology that Galton was proposing is one that is very similar to that of contemporary genetics. Namely, to reduce complex characteristics or behaviours to predetermined inherited causes, to find those individuals with particularly poor scores for these complex behaviours, and to select these individuals for some form of intervention which will help to improve their lives and also to improve society. The methodology may be similar, but the underlying philosophy is different. At the heart of the contemporary methodology is a desire for social welfare and to improve the lives of individual people. Galton also invented the terms nature and nurture. He called them a convenient jingle of words, although he was not to be aware of the darker controversies and misconceptions that these terms would later produce. Galton's approach to studying human behaviour, and also the approach of his successor Pearson, were to use statistics to study the differences between individuals in a population. In doing so, they laid the foundations for a lot of contemporary social science research, because theirs is the approach that developed the ideas of standard deviation, regression, and the population level analysis of characteristics. This is one of the reasons why Galton's legacy is so very uncomfortable today. Many generations of researchers have used his principles without necessarily agreeing with his methods, and certainly not agreeing with his social philosophy. Galton had no clear idea of how inheritance took place. He had no mechanism underlying his thinking. He relied on the ideas of his famous cousin, Charles Darwin, who had put forward the mechanism for inheritance known as pangenes. Unfortunately for Darwin and Galton, this idea was incorrect and was not able to be developed further. At the same time that Galton was debating hereditary genius in the sophisticated gentlemen's clubs of central London, Gregor Mendel, an Augustinian monk with a lifetime vow of celibacy and poverty, was trying to understand the mechanisms behind selective breeding. Mendel proposed that the differences between characteristics were controlled by factors that were inherited from parents to offspring. These were later called genes. Mendel proposed that these factors could be different, and the differences could lead to the differences observed between the characteristics. For example, between purple and white flowers in peas. Mendel proposed that pea plants have two of these factors, whereas their gametes, pollen grains or eggs, only have one. At fertilization, the factors inside the gametes come together to produce the characteristics seen in the offspring. A generation of pea plants produced from a single cross would have predictable ratios which could be observed and counted. It was Mendel's genius that allowed him to predict these observations and then carry out the necessary experiments which showed that the predictions were correct. Galton's work was largely descriptive. Mendel's work was more analytical because he did provide a mechanism to explain the observations. This is why Mendel, and not Galton, is said to be the father of genetics. Mendel published his ideas in a single paper in 1866. in an obscure journal, and his thinking was largely ignored until 1900, 17 years after his death. In the early years of the 20th century, there appeared to be two rival schools of thought for the emerging science of genetics. The conceptual ideas of Mendel, which could be tested experimentally, immediately had wide implications for plant and animal breeders who wanted to improve their crops and livestock. This was countered by the school of Galton and Pearson, the biometrical school, which insisted on accurate measurements and statistical analysis as the basis for drawing their conclusions. For nearly 20 years, these ideas were regarded as being in opposition to each other, and also in opposition to Darwin's theory of evolution. It wasn't until the end of the First World War in 1918 that Darwin's theory of evolution, Mendelian genetics and biometrical genetics were reconciled. And they were brought together by an important biologist called Ronald Fisher. Fisher showed mathematically how Mendel's factors, which were now called genes, could be extended for complex characteristics which were controlled by larger numbers of genes, each having a very small effect on the characteristic. He called this reconciliation biometrical genetics. In doing so, he showed how biometrical genetics was reconciled with Darwin's theory of evolution. Fisher's seminal 1918 paper invented a new key term, that of heritability. Understanding the strengths and limitations of heritability will be at the heart of this course. So, what does all of this history mean for people today? It's now time to explore how genetics impacts people's lives today.